Type 1 hypersensitivity reaction is also called as the immediate type of reaction and involves immunoglobulin IgE mediated release of the antibodies against a soluble antigen. So in this type of hypersensitivity reaction, when the antigen enters into your body, that causes activation of the B cell or our immunity system to release antibodies. Which type of antibodies? IgE antibodies. So hypersensitivity type 1 reaction is mediated by the antigen and IgE antibodies. This results in mast cell activation or degranulation and the release of histamine and other inflammatory mediators. So once the antigen enters into your body, that causes activation of immune system, then there will be release of IgE antibodies, and IgE antibodies will go and bind with the mast cell, activate the mast cell and cause degranulation of the mast cell. So the mast cell will release histamine and other inflammatory mediators. The eosinophil also play a role in hypersensitivity type 1 reaction. Interleukins that play a role, they are interleukin 4 and interleukin 5, and these interleukins, they cause activation or migration of the eosinophil and release of the IgE antibodies by activating the B cell. Now let's go for the diagrammatical image so you can betterly understand that how the mechanism of the hypersensitivity reaction works. You know that the first one, upon first exposure to the allergen, the antigen presenting cell processes the antigen and presents it to the T helper cell. In first lecture, I already said about the antigen presenting cells, the mechanism of antigen presenting cell. Here they are saying that whenever the macrophage, it engulfs the antigen, it processes the antigen and present the antigen on MHC class 2 molecule, and there will be interaction of T cell receptor present on the helper T cell, and this receptor is called as the antigen activation of the T helper cell. Then there will be release of different sort of interleukins like interleukin 4, interleukin 12 by the T helper cells. These interleukins causes activation of the B cell and differentiation of the B cell. Now the B cells, there will be proliferation of B cell. You can see in the third point, there will be B cell proliferation and differentiation into plasma cells. What is the function of plasma cells? They synthesize and secrete IgE antibodies. So these are the plasma cells. Now they are releasing IgE antibodies. Now IgE antibodies, they act on the mast cell, and all these granules are the preformed granules of histamine inside the mast cell. So IgE binds to the mast cell by Fc region, sensitizing the mast cell. So from 1 till 4, it is a phase of sensitization. After sensitization, there will be release of a lot of histamine and other inflammatory substances, and that shows different sort of changes in the body. There will be vasodilation, bronchoconstriction. So upon subsequent exposure to the allergen, the mast cell with IgE bind to the antigen and release inflammatory molecule, result in allergic symptoms. So this is the effective phase or effector phase, sensitization phase and effector phase. 1 till 4. Here the same images here, the effector phase and the sensitization phase, but they want to show us some symptoms, like when there will be release of the mast cell and other cytokines. There will be smooth muscle contraction, bronchodilation or dilation of the blood vessels, hypersecretion by the mucous glands, platelet aggregation, sensation. Now we are going to talk about the type 2 hypersensitivity reaction. Type 2 hypersensitivity reaction is also known as antibody dependent cell mediated antibodies, leading to complement system activation and cell damage or the lysis. So how this process happens? You know, for example, this is a cell and antibody, and there will be receptor. So this antibody will come and bind with the receptor. Then there will be activation of complement system, and this complement system destroy the cell. So for this, we need to have a cell, we need to have antibody and a complement system. Then there will be destruction of the cell. That's why it's called as the antibody dependent cell mediated cytotoxicity. So diagrammatical presentation. So the diagrammatical presentation will show you two mechanisms. Number first, complement mediated, and number second, natural killer cell involvement. This is the antibody dependent cell cytotoxicity. So in the first one, what's going on? Here is the target cell. These are the antibodies. Now antibodies, they attack on the target cell. Then there will be activation of different sort of complement system. Then the complement system, these are the complement systems, and they cause lysis or osmotic lysis of the cell. How? I said this is the cell, this is the antibody. We have a complement 4, 3, 2, 5, 6, 7, 8 and 9. So 5, 6, 7, 8, 9, they are the membrane attacking complex that attack on the membrane, make the membrane porous, water will go inside and there will be lysis of cell. Among these, C3b that cause opsonization. What is opsonization? This C3b will call macrophages and help to destroy the cell. Same here. So this one is the macrophage, this one is the effector cell or the target cell, this is the antibody, and here bind the green one, they are the C3b complement system. So the C3b, it allow the macrophage to come more rapidly and to engulf this cell. You can see here. So there will be a phagocytosis by the macrophage, and this is called as opsonization. So number first, because of complement system activation. Number second, there will be the target cell, antibodies directly, and no involvement of complement system, no complement system, then what are the cells that are responsible to destroy the cell? They are the natural killer cell. They have granules inside, and these enzymes will release and cause destruction of the cell. So this natural killer cell will bind the antibody and directly kills the cell. So here, and the third one, they also give another one also, that is anti-receptor antibodies. The anti-receptor antibodies, they will go and activate the receptor or inhibit the receptor. If you talk about myasthenia gravis, there will be inhibition of the receptor, and if you talk about the grave disease, then there will be activation of the receptor. In this condition, given the myasthenia gravis, where all these blue, they are the receptors for the acetylcholine that causes muscle contraction. So when the synaptic knob or the end of the neuron, they releases a lot of acetylcholine. These acetylcholine needs to act on the acetylcholine receptor, but they are blocking by these autoantibodies. So there will be no action of acetylcholine, and there will be no or very few contraction of the muscles. So it's inhibiting the receptor, and this is antibody mediated, and this is an example of type 2 hypersensitivity reaction. Now let's go for the type 3 hypersensitivity reaction. Type 3 hypersensitivity reaction. Type 3 hypersensitivity reaction is also known as immune complex reaction. It also involves antibody IgG and IgM and sometimes IgA antibodies. The buildup of these immune complexes that results in complement activation. Again, there will be activation of complement system, which leads to polymorphic leukocyte chemotaxis and eventually causing the tissue damage. So here what happened? Last time we said that we have antigen and antibodies. They bind, they activate certain other cells, then there will be destruction. And the antigen enters into your body. This antigen binds with the antibody, and this antigen-antibody complex circulate in our blood vessel, in our circulation, and it will go and land up at any place. If I give you an example of glomerulonephritis, then these antibodies will go and attack to the endothelial cell of the glomerulus. Once this antibody and antigen complex will bind or get stuck to the endothelial cell, then there will be again activation of the complement system. Then this complement system causes damage of the endothelium. So this is type 3 hypersensitivity reaction, when there will be activation of when there will be a formation of antigen antibody complex that will go and deposit it anywhere, that cause activation of the complement system. And they said that there will be chemotaxis of the polymorphic nuclear cells. So because of C5a is a complement system, one of the complement. This cause attraction and migration of neutrophils. They call the neutrophils. So this is what chemotaxis. Previous we said C3b helps in opsonization and C5a helps in the chemotaxis. So in this particular image, the antigen comes and bind with the B cell, which is antigen presenting cell, cause activation of plasma cell, formation of the antibodies. These antibodies will then go and attack with the antigen, and there is a formation of immune complex. This immune complex will deposit it at basement membrane or the endothelial cell, and there will be activation of complement system, and some complement system like C5a causes activation and chemotaxis of the neutrophils. There will be neutrophil activation, and along with the neutrophil and the complement system, they both causes tissue damage. So examples, serum sickness, Arthus reactions, systemic lupus erythematosus. These are the examples of the type 3 hypersensitivity reaction. Now move forward for the type 4 hypersensitivity reaction. The type 4 hypersensitivity reaction is a delayed type of reaction. It will take the pathogenesis. So it also have two phases, sensitization phase and effector phase, like the type 1. So when the bacteria or any antigen comes inside the body, the antigen presenting cell like macrophages, they engulf, present the antigen on the surface, present to the T cells. T cell releases different sort of cytokines, T helper cells. Those cytokines will act on the T helper cells. They are the auto cytokines. These autocytokines act on the cell, causing proliferation and differentiation of usually proliferation of the T cells. And now the T cells, they release interferon gamma, tissue necrotic factor beta. They act on the on the macrophage. Now the resting macrophage will become the activated macrophage, and now these T cells as well as the activated macrophages, what they do, they will increase the response and causes a local inflammation along with the formation of giant cell granuloma. So they said antigen presenting cell, then there will be T cell activation, delayed type of hypersensitivity cell. T helper cell produce interferon gamma, tissue necrotic factor alpha beta, interleukin 2, interleukin 3, interleukin 8, and certain other IMF, MCMF, Then there will be activation of macrophages, MHC. Type 1 hypersensitivity reaction, type 2 hypersensitivity, type 3 and type 4 hypersensitivity reactions. Type 1, allergen, IgE, mast cell and degranulation, histamine. Type 2, antibodies, antigen presenting cell, binding to the T cells, natural killer cell, or there will be activation of complement system by the antibodies. Type 3, formation of antigen antibody complex, attraction or chemotaxis of the neutrophil. Type 4, activation of macrophages and T cell, both help in the formation of granuloma. IgG mediated cell cytotoxic hypersensitivity, type 2. Immune complex mediated hypersensitivity is a type 3. Cell mediated type hypersensitivity is a type 4 hypersensitivity reaction. So IgE is bound to the mast cell via its Fc portion. When an allergen binds to these antibodies, cross-link IgE induce degranulation of mast cell. What is the cause? Systemic and localized anaphylactic reactions, seasonal allergies, including hay fever, food allergies, such as those to shellfish, peanuts, hive, and eczema. So seasonal allergies, anaphylactic reactions, eczema, allergy to food, allergy to certain other allergens is a type 1 hypersensitivity reaction. Type 2, cells are destroyed by bound antibodies either by activation of the complement system or cytotoxic T cells, and FC receptor for the antibodies with an FC receptor. So red blood cell destruction or destroyed by the complement system and antibody during the transfusion of mismatched blood or during erythroblastosis fetalis. These are the example of the type 2 hypersensitivity reaction, like destruction of the RBCs because of the antibodies, and this antibody activate the natural killer cell or the complement system that causes destruction of the cells. Type 3 hypersensitivity reaction is antigen antibody complex. They are deposited in tissues, causing activation of the complement system, which attract neutrophil to the site and causes damage to the site. Most common form of the immune complex diseases: glomerulonephritis, rheumatoid arthritis, systemic lupus erythematosus. These are the example of the type 3 hypersensitivity reaction. Type 4, mediated by T helper cell that secreted cytokines which activate the macrophages and cytotoxic T cells and can cause macrophage accumulation at the site. Most common form are contact dermatitis, tuberculin reactions, autoimmune diseases such as diabetes type 1, multiple sclerosis, and rheumatoid arthritis. So this is the example of the type 4 hypersensitivity reaction. Some reactions they have both type 3 and type 2, sometimes type 3 and type 4. They have both like that. So thank you so much for listening.